In this lecture, I'm going to talk about the important aspects of the first three pieces of cellular respiration: glycolysis, pyruvate oxidation, and the Krebs cycle, or it is called the citric acid cycle as well. Krebs is the name of the guy who discovered the cycle, or who described the cycle, but it's also called the citric acid cycle. Glycolysis is going to happen in the cytoplasm. That's here. Pyruvate oxidation is going to happen in the matrix of the mitochondrion, and the Krebs cycle is also going to occur in the matrix of the mitochondrion. We'll go through each step one at a time and talk about, most importantly, the input and output of these cycles. Remember that our goal is to take glucose, the starting substrate in this process, and ultimately get out ATP. We're going to strip apart the bonds of glucose, release the energy in there, and put it into ATP. The first three phases of cellular respiration strip all that energy away from glucose and primarily store it as NADH. And NADH will then be used to make ATP. However, there is a little bit of ATP production in this process as well, and so we'll stop at each step and kind of summarize what's happened. Glycolysis is our first stage, and the input and output is probably most important about glycolysis. Glucose is ultimately, through a series of 10 reactions, converted to two molecules of these 3 carbon compounds called pyruvate. 3 carbons in pyruvate, 6 carbons in glucose. So by the end, we're going to end up with 2 times 3, 6 carbons still included, but 2 molecules of 3 carbon pyruvate. And along the way, we're going to strip some of the energy to make a little bit of ATP, but importantly, make some NADH as an energy carrier to later make ATP through electron transport. Glycolysis is split into 2 phases: the energy investment phase and then the energy payoff phase. The first 4 reactions are the energy investment phase, and so it actually ends up taking a little bit of ATP to get glucose activated to be able to then take it through reactions to extract the energy from glucose. So within the first 4 steps, there are 2 steps where we have to actually hydrolyze ATP, and notice we put little phosphate groups on here. That puts them into a situation where enzymes later can act on these molecules and extract even more energy than we put in. Every arrow here represents a chemical reaction, and every arrow represents a place where an enzyme acts. The enzyme that acts right here only affects this substrate. It takes ATP and phosphorylates this particular substrate to make something new. So every step is an enzyme catalyzed reaction, and every molecule at the end of a step is what's produced from the reactant before the enzyme acts. So by the end of the energy investment phase, notice that we have put in 2 molecules of ATP, and we've also split glucose into 2 3-carbon molecules called G3P, glyceraldehyde 3-phosphate, but these aren't the end products of glycolysis. Glycolysis then is a series of chemical reactions that convert these 3-carbon molecules into pyruvate. One of these is actually something different initially, but it's converted to glyceraldehyde 3-phosphate. That's why we say there are 10 reactions when only you see 9 here. But what's important is in these last few reactions, there are 5 described here, we get out ATP. And notice for each one of these 3 carbons, we get out 2 molecules of ATP. So we're going to end up netting 2 molecules of ATP. We put 2 in, but we get 4 out. Also important, we take some of the hydrogen that was part of these molecules and we're going to attach it to NAD+. And NADH now is carrying high energy electrons to be used later. This is a redox reaction, that oxidation and reduction process. NAD+ gets reduced, this molecule gets oxidized. It loses some hydrogen. We don't see where all the hydrogen gets lost because that's really not so important as just knowing that we took some of the high energy electrons and put them onto NAD+. So by the end of the energy payoff phase and by the end of glycolysis, we have 2 molecules of 3-carbon pyruvate for that original glucose. We also net 2 ATP. We also net 2 NADH. Notice that the process is gone through twice because now we have 2 3-carbon molecules for every 1 original glucose. The ATP production that we saw in glycolysis is a result of something called substrate level phosphorylation. Notice that I had a phosphorylated substrate here. When the reaction occurred, I got ATP out and I've lost a phosphate here. The reason why this process right here is called substrate level phosphorylation is because the enzyme's substrate had a phosphate, and one of the other substrates for the enzyme was ADP. I phosphorylated, attached a phosphate group to ADP from a substrate, and so that is substrate level phosphorylation. The phosphate was attached to ADP. What's released is an dephosphorylated product, an ATP. Substrate level phosphorylation is a type of ATP production that we're going to see in the citric acid cycle as well. But let's talk about what happens to these pyruvate molecules first. Pyruvate, and also the NADH that was made here, is going to be shuttled into the mitochondrion, all the way into the matrix. At that point, we're going to have pyruvate oxidation. And in pyruvate oxidation, the pyruvate molecule gets decarboxylated, so we actually lose a carbon, and we lose hydrogen to make more NADH. And as a matter of fact, we make 2 NADH because we're putting in 2 pyruvate. The 2 pyruvate came from the 1 molecule of glucose. In addition, there's this crazy little addition here called coenzyme A, and coenzyme A is attached to pyruvate to make this now 2-carbon, because we stripped a carbon away, acetyl-CoA. And the only reason why we care about coenzyme A is because it prepares our now 2-carbon molecule for entry into the Krebs cycle. And the Krebs cycle, or citric acid cycle, is where we're going to finally strip apart everything that used to be glucose. But I want you to notice that what we got out of pyruvate oxidation is 2 molecules of acetyl-CoA, 2 molecules of CO2, and 2 molecules of NADH. And the reason why we have 2 of everything is because we put in 2 pyruvate for the 1 original glucose. NADH is produced twice for every 2, because of 2 molecules of pyruvate. CO2 is produced twice, 2 coenzyme A are put in, and ultimately we get 2 acetyl-CoA to go into the Krebs cycle. So pyruvate oxidation is our second step of cellular respiration. Next is the Krebs cycle, or the citric acid cycle, and our acetyl-CoA is going to go through a series of reactions that ultimately produce 2 molecules of ATP, because there are 2 molecules of acetyl-CoA going through this, 2 CO2 for every 1 acetyl-CoA, so 4 CO2, 3 NADH for every 1 acetyl-CoA, so 6 NADH, and then another molecule which is called FADH2, which is just analogous to NADH. There'll be 2 of those produced because we're putting through 2 acetyl-CoA molecules. Now it's a lot more complex than this, but I just wanted to give you a summary of the input and output first. All of this is happening in the mitochondrial matrix, and remember, we went through glycolysis, we went through pyruvate oxidation, and now it's time for the Krebs cycle. And the Krebs cycle is a series of chemical reactions that produce all of those outputs that I've described for the Krebs cycle, or the citric acid cycle. Let's go through these and through the process in a little bit slower steps. First, acetyl-CoA is joined to a molecule that has 4 carbons. Ultimately, what we're going to have is we're going to have a 6-carbon molecule regenerated. But remember, just 2 of these came from glucose. CoA is released, and we're going to end up with a molecule called citrate. Citrate or citric acid is the basis for the name of the citric acid cycle. What we've done is we've prepared these little 2 carbons that came from glucose to go through the rest of the cycle. Citrate is immediately converted to another molecule, and notice we lose some CO2. We also produce NADH. It's important to remember that for every step here, really it's happening twice for every 1 molecule of glucose, because we've split glucose into pieces. We get NADH and CO2 throughout these steps, and also 1 molecule by substrate level phosphorylation of ATP. This is all a bunch of redox reactions, reduction of NAD plus to be NADH, and oxidation of what used to be glucose. So we are slowly but surely oxidizing, stripping away electrons from what used to be glucose. Everything that we needed to get produced, for the most part, is produced here, with the exception of some FADH and NADH. We're going to put a little bit of water in, which is not important for us, and ultimately we're going to regenerate oxaloacetate, that 4-carbon molecule, so it can keep on being joined to acetyl-CoA. Most important is that we've reduced a bunch of NAD plus and FAD so that we can then take these high energy electrons to the electron transport chain. Each one of these arrows is an enzyme catalyzed reaction. This, for example, succinate, was a substrate for this chemical reaction, as was FAD. The result, or the product, a 4-carbon molecule, and FADH2. So every place here requires an enzyme. So by the end of these 3 phases, this is what we've produced from 1 molecule of glucose. In glycolysis, we net 2 ATP, 2 NADH. The ATP can go and be used to drive energy-requiring steps in the cell. Pyruvate oxidation produced some CO2 byproduct and NADH. The Krebs cycle produced some more ATP, which can be used, 6 NADH, and FADH2 and CO2. So ultimately, we know that we've gotten rid of all the carbons that used to be glucose and put them into CO2. And all of the NADH and FADH2 can now be shuttled to electron transport. And I just noticed that this should actually say 2. These 2 should both say 2. Because for every 1 round of the Krebs cycle, we get 1 ATP and 1 FADH2. But remember, we've gone through it twice, which I've actually counted for everything else. We have 2 NADH from glycolysis, 2 NADH from pyruvate oxidation, 6 NADH from the citric acid cycle, 2 FADH2 from the citric acid cycle. And all of these are going to be shuttled to electron transport to make a bunch of ATP. CO2 that we got from these 2 phases is now a byproduct, and that'll be just exhaled and we'll get rid of it. The ATP that's produced by substrate level phosphorylation can be used to do things in the cell, but we still have to make the bulk of our ATP through electron transport, and that is the subject of the next lecture.